Perfect.
Okay. Well, good morning or good afternoon, depending on where you are. I'll be making one of 4 presentations this meeting about evaluation of the U.S. peanut Minicore for response to water deficit stress in 3 states, Texas, Oklahoma, and Virginia. The reason for this is that water deficit is considered to be the single largest cause of yield loss in peanut. In the Southwest, peanut is grown, Southwest United States, peanut is grown exclusively under irrigation because our rainfall is considerably lower than in other regions. Typically in the Lubbock area, we may get 12 to 16 inches, let's say 300 to 400 millimeters of rainfall during the growing season. This figure over at the right shows that in the West Texas, New Mexico, Oklahoma area, where peanut is grown, the levels of aquifer are being depleted quite rapidly, and so improved water deficit tolerance is needed for growers. There have been a number of publications that over the last couple of decades that have demonstrated that there's substantial genetic variation for responsive peanut to water deficit using a number of different measures. And we would like to use these for a trait-based selection because that would allow breeders to combine different components of response to water deficit together in a variety. However, many of these measurements are very tedious to make and really not feasible to use in phenotyping a large breeding population. And therefore, we really need high throughput phenotyping as well as DNA markers that can be used for selection. Now, previously I presented that we had screened the Minicore and identified some accessions that were useful, at least based on Texas data, and done some SSR marker work and identified a limited number of SSR markers from a fairly limited screen of the genome, and validated these markers in one population where it showed that 3 of these markers gave a 14% yield increase compared to no selection based on markers. However, in another population, there really was no significant effect. We attribute this to the fact that the genome coverage of the markers we had was fairly limited. The other issue we've seen is that in some of our materials, while we have good yield response, the grades are generally fairly poor. We attribute this probably due to bringing low grades from the Minicore accessions. And so we really wanted to go back and revisit this. Hypotheses are the U.S. peanut Minicore collection has significant genetic variability for response to water deficit. We wanted to test this across 3 states. And goals in that identify accessions with superior yield and grade and take ground-based measurements that can give us some idea of the different responses. Set 2 other hypotheses that what I'm presenting is really preliminary to 3 other presentations being made are that high throughput phenotyping methods will make screening large populations feasible. And for this, identify a limited set of accessions that can be used for this purpose and for more detailed physiological work. And that high throughput genotyping, GWAS using SNP-based markers can identify markers for breeding program. And again, the second and third hypotheses, others will be presenting, which I'll mention a little later. So the work that was done was planting the Minicore, 3 locations in 2017, planting in early to mid-May in Virginia, Oklahoma, and Texas, and in 2018 also in Virginia. Experimental design was a randomized complete block design with 3 to 4 replications depending on location. In irrigation, water deficit, we're looking for mid-season drought, and therefore early in season before flowering, we applied water to 75% of evapotranspiration replacement, and at mid to late season also. During the mid-season drought, generally we applied no water except in Texas where this actually was necessary just to keep our experiment alive. The common canopy measurements we took were listed here. And we had multiple readings taken June, July, and August. The materials were harvested in October, roughly 145 to 160 days after planting, depending on location. And the analysis I'll present are based on mixed models using Systat and Fisher's protected LSD test. In Virginia, the material was planted at the Tidewater Research Center, and you can see there's a kind of a white sugar sand soil, which doesn't hold water very well. And this shows 2017, progressive drying of the soil until a major rainfall event early August. It was also planted in Texas at the Lubbock USDA ARS. We had severe water deficit stress. Our goal was irrigation of 25% ET replacement in mid-season. In fact, we had to supply additional irrigation to keep the experiment from dying. And we had biweekly ratings. And this just shows an example of some of the ranges that we saw for different traits, and that these were all highly significant among differences among accessions. And in Oklahoma, experiments were planned at the Oklahoma State Fort Cobb Research Station. Again, mid-season was under rain-fed conditions. Due to the size of the experiment, it was not possible to plant the material under rain-out shelters. And so mid-season, we had to rely on basically what rainfall there was. And they also took some LIDAR measurements, which I won't be presenting here. So the analysis, what we see is that for the common measures, in fact, these were all highly significant, different among accessions. And in some cases, there were highly significant differences among locations. In some cases, there were also significant location by genotype interactions. And I want to present two cases here. One is the SPAD chlorophyll or SCMR readings, borderline significant for a location by genotype interactions, highly significant among accession. And in blue, what we have is accessions that were in the top values, red the lowest values in the test. And what we can see among the locations is generally those accessions that did well on average did well in all locations. So there's a pretty good correlation among locations. Now for yield, on the other hand, this wasn't necessarily true. What I show here, so we had 3 locations in 2017, 1 in 2018, and I show those. And so what I show down here is there are very major differences in yield among locations. I see in Texas, 907 pounds per acre, about 1,000 kilos per hectare due to extreme water deficit. In Oklahoma, 3,300, Virginia, 4,800 and 2,500 due to more rainfall. And what we have here is the top 10% of the accessions in Minicore, the bottom 10% for yield. And what we see is two things. One is a number of accessions did as well as, or maybe even better than checked varieties. 2, there were some accessions that did well for yield at all locations, but other ones here that did well under the extreme drought in Texas did not do very well in Oklahoma or Virginia. So we are seeing location-specific differences in yield. What I show here is the same accessions, the top 10% in Minicore, bottom 10% in Texas. And what I've colored in blue is the favorable accessions, red the unfavorable ones or responses. In general, the highest yielding materials had high flowering counts, generally pretty high SPAD chlorophyll ratings with a couple of exceptions. A little bit surprising, tended to have higher canopy temperature instead of lower ones. And also then we see some higher leaf closure during the heat of the day to conserve water, and generally somewhat larger canopy with maybe a proxy for canopy coverage here. Correlations by location, what we see is in green, this positive correlations, red or orange, the negative ones. In Texas, the highest correlations were yield with flowering, leaf closure. In Virginia, yield with NDVI and canopy width, and in Oklahoma, again, with leaf closure and canopy width. Across all locations, the highest correlations with yield, positive with SPAD chlorophyll ratings, leaf closure, canopy width, inverse correlations with wilting and plot height. I tried taking these data and putting them in a stepwise regression to see if we can make any models, and yes, there were. In Texas, what we saw is the biggest contributions in yield in this model were flowering, SPAD chlorophyll reading, canopy temperature, and measures of canopy coverage. In Oklahoma, it was actually leaf closure at the 2 different digging dates, and also flowering and canopy coverage. In Virginia, wilting and NDVI were the strongest, along with canopy coverage in both years. What we can see is that when we compare this, we could explain about 25 to 50% of the phenotypic variation using the trait-based measures. Looking at yield and grade, we had several accessions that did fairly well, relatively speaking, nothing graded over 68%, and some varieties graded 48 to 52%. So grades were fairly low under water deficit. Yields, of course, were fairly low. There were some Minicore accessions that statistically were very similar to the best varieties. Some were as good as or even better than some of the check varieties. So we conclude that there are some Minicore accessions that have yield and grade that we think would be useful in a breeding program. All the measures I've presented so far are above ground measurements, but roots are a very significant part of the plant, but we have no way of phenotyping these efficiently. So actually, Dr. Dirk Hayes came up with some of his graduate students and brought a ground penetrating radar unit and tested this in the field. They did this late in the season when there were near harvest. And what they did is then use different radar wavelengths and different times, that is essentially depths, or look at different depths of the echoes, did statistical permutations on this and came up that they could explain about 50% of pod yield based on the radar echoes. If they refine the parameters to different market types, they could come up with higher correlations. So this is promising. What I would hope in the future is that mid-season it might be able to do testing and measure root biomass, which should give us a way of measuring that. So I just measure future work, at least beyond what I'm presenting here. One is GWAS. In fact, this is the idea is we would like to have molecular markers for this. So we cataloged the materials we had. Due to the timing of this experiment, we had to take materials that we already had in Texas and Oklahoma. And Texas and Oklahoma ARS programs have evaluated the Minicore over a number of years, and we previously purified these. And so most of the accessions are similar to what Charles Chen has worked on, but we've noticed some differences. Therefore, we extracted DNA from this separately and sent this off for genotyping. And Joy Sung had presented this results of GWAS in the graduate student competition yesterday. You could see this online. And for selections for high throughput phenotyping, we selected a limited number of accessions to represent the diversity we saw in the Minicore, so that this test could be grown under rainout shelters for more detailed and precise phenotyping. And in some of the results of UAV and rainout shelter data will be presented by Maria Belota tomorrow, Rebecca Bennett in her poster session. And what I say is we took 22 Minicore accessions plus 6 checks by subspecies to make contrasting high and low values for the different traits that we had here. And finally, then, sort of missed the summary. And so summary, what we have is that our hypothesis of large differences among accessions for traits was accepted. We saw large differences for all the traits that we measured. And some of these accessions appear to be very useful, have good values for both yield and grade. Rankings for some traits were consistent across locations. For yield, no. High throughput phenotyping, we made selection of accessions and got the ground-based data that could be used for comparison. And for GWAS, collected the DNA and had physiological data to compare against as well as for yield. Overall summary, we concluded the Minicore accessions have a potential for use as parents for yield and grade under water deficit. However, it would be best if we had high throughput phenotyping and marker data that we could use for this work also. And finally, just want to acknowledge this work was supported in large part by a NIFA grant. Maria Belota is lead PI, and we've also received additional funding support for other aspects of this work that have been presented here. And so that is all that I have to present. And I guess we go back. If there's time, I can take some questions.
There are a couple of quick questions. And so the first one is from Chris Butts. The ground penetrating radar was detecting pod load since it was late in the season?
Yes, that is correct. They felt that since this hadn't been attempted in peanut before, they had a better chance of detecting pods simply due to a much greater mass of pods underground than roots.
Yeah, I thought that was very, very ingenious. And then the second question, quickly from Bryce Casneve, would machine learning allow to differentiate roots versus pods when using the ground penetrating radar?
I don't know. I haven't done the experiment. They tried explaining what they did, and a lot of it was over my head. But I would hope that it might be possible, or perhaps to take the root mass data mid-season where we wouldn't have much for pods, but it would be mostly roots.
Sure, sure. Makes sense. Well, Mark, thank you very much for your presentation.